Hello, welcome to Study Hub Pro. Today, we're diving into one of the most fascinating tricks in the microbial world, conjugation. It's how bacteria, even though they don't have sex in the way we think of it, can still share genes. And this little trick gives them an unbelievable ability to adapt and survive.
So let's kick things off with a bit of a puzzle. Bacteria, they usually just reproduce by splitting in two, making perfect clones of themselves. So if that's all they're doing, how in the world do they evolve so quickly? Where does all the variation for things like antibiotic resistance come from? Well, this incredible image right here gives us a huge clue. See that tiny little bridge connecting those two E. coli cells? That's called a sex pilus, and it is the key to a form of genetic sharing that pretty much changes everything.
The answer is a process we call horizontal gene transfer. You can think of it as sharing genes sideways with your neighbors instead of just passing them down to your children. Now, bacteria have a few different ways of pulling this off, but today we are laser focused on the most direct method, conjugation. It's basically a literal cell-to-cell handshake to swap some DNA.
But how did we even discover this was happening? I mean, you can't just see it with a regular microscope. To really get it, we've got to go back in time and look at the seriously clever experiments that first proved this was even possible.
Okay, first things first, we need to get two key terms straight, auxotroph and prototroph. The easiest way to think about it is this. An auxotroph is like a picky eater. It's missing the recipe, that is the gene, to make a specific nutrient for itself, so you have to add it to its food or it won't grow. A prototroph, on the other hand, is totally self-sufficient. It has all the recipes it needs to make everything from scratch. This simple difference, well, it's the whole secret behind the brilliant experiment we're about to look at.
So this is the classic 1946 experiment by Lederberg and Tatum. What they did was they took two different strains of these picky eater E. coli. Strain A couldn't make two nutrients, and strain B couldn't make three different ones. Now, on their own, neither of them could survive on a really basic minimal nutrient plate. But, and here's the magic, when they mixed the two strains together, some colonies started to grow. These new bacteria were prototrophs. They were self-sufficient. The only way that could have happened is if the two strains somehow got together and shared their genetic recipes, creating a brand new, complete cookbook.
That, of course, was the next big question. Did the bacteria have to physically touch each other to swap genes, or were they just releasing some kind of helper molecules into the liquid that the other bacteria could slurp up? Science is all about ruling out those other possibilities.
And this incredibly clever YouTube experiment by Bernard Davis settled the debate for good. He put the two strains in each arm of this U-shaped tube, but he separated them with a filter. Now, the filter had pores that were small enough to let the liquid mix, but too small for the bacteria to pass through and touch each other. And when he tested the bacteria, nothing. No gene transfer, no growth on the minimal plate. That was the proof positive. No physical contact, no gene swapping. Period.
So, we've established that they absolutely need to make physical contact. Now, let's actually dive into the mechanics of how that transfer happens. And we'll start with the most basic version of it all. All right, the absolute star of this show is a little piece of circular DNA called the F factor. The F stands for fertility. It's a type of plasmid. Now, what's really important is that it's also an episome. All that means is that it has two options. It can either live on its own, just floating around in the cytoplasm, or it can actually integrate itself into the main bacterial chromosome. This F factor carries the genes for making that sex pilus we saw earlier. A cell that has this plasmid is called an F+ cell, and it's the donor. A cell that lacks it is called F-, and it's the recipient. Here's a quick snapshot of the whole process. You've got the donor cell with the F factor. It connects with the recipient, and by the end of it all, they both have a copy. Pretty neat, right? Now let's break that down step by step. So it really happens in a few key steps. First, the F+ donor cell sends out its pilus and makes contact. Second, that pilus retracts, pulling the recipient cell in really close, forming a bridge. Third, the donor starts copying its F factor plasmid using this cool rolling circle mechanism, and it feeds one strand of that DNA into the recipient. Once that single strand is inside the recipient, it's used as a template to make the full double-stranded copy. And what's the result? The original F- recipient is now converted into an F+ donor. The really important thing to remember here is that in this basic type, it's only the plasmid that gets transferred, not the main bacterial chromosome. But what happens when that F factor isn't just floating around freely? Remember how I said it was an episome and could pop into the chromosome? Well, this is where things get really, really interesting.
When that F factor does integrate itself into the main bacterial chromosome, we call that cell an Hfr cell. It stands for high-frequency recombination. It's still a donor cell, absolutely. But now the F factor isn't a separate little thing, it's a part of the cell's main genome.
And this integration completely changes the game. When an HFR cell decides to conjugate with an F- cell, the transfer process still starts within that F factor sequence. But because the F factor is now physically attached to the chromosome, it acts like an engine and begins to drag the entire host chromosome along with it into the recipient cell.
Now here is the absolute key detail, and this is a classic exam question, so listen up. Transferring an entire bacterial chromosome takes a long time, like about 100 minutes. That little physical connection between the cells is pretty fragile, and it usually breaks long before the whole transfer is finished. And since the F factor itself is split, with the beginning part going in first and the very end of it going in last, the recipient cell gets a big chunk of chromosomal genes, but it almost never gets the full F factor. So what does that mean? It means it picks up new genes, but it stays a recipient. It remains F-.
Okay, on to our third and final type. Sometimes an F factor that was integrated into the chromosome decides to pop back out and become an independent plasmid again. But this process, called excision, can be, well, a little bit sloppy. Sometimes when it leaves the chromosome, it accidentally grabs a few adjacent chromosomal genes and takes them along for the ride. This new modified plasmid is called an F' plasmid, and the cell carrying it is an F' cell.
So you can imagine what happens next. When this F' cell connects with the recipient, it transfers that entire special plasmid, F factor, and the extra bacterial genes it picked up. The recipient now has a second copy of whatever chromosomal genes were accidentally grabbed. We call this state a partial diploid, or a merozygote. And you can see how this becomes a super efficient way to spread very specific traits, like, say, an antibiotic resistance gene, through an entire population of bacteria.
Now, this ability of HFR cells to transfer the chromosome in such an orderly, linear fashion wasn't just some biological curiosity. It actually gave scientists an incredibly powerful tool to do something they had never been able to do before, to actually map the bacterial chromosome.
The technique they came up with is called an interrupted mating experiment. And honestly, it is pure genius. Here's what you do. You mix an HFR strain with an F- strain and just let them start conjugating. Then, at specific time points, say after 5 minutes, 10 minutes, 15 minutes, you take a sample and you literally toss it in a kitchen blender for a few seconds. That shearing force is just enough to break those delicate little conjugation bridges without killing the cells. Then you plate the recipient cells and check to see which new genes they've received.
And what you get are results that look just like this. Genes that are closer to where the F factor inserted, the origin of transfer, get transferred earlier. So you might see that the ASI gene gets there in 8 minutes. The TON gene shows up at 10 minutes. The LAC gene at 17, and so on. By checking which genes have arrived at which time point, you can figure out their exact linear order on the chromosome and even the relative distance between them. It's like creating a map based on travel time. It's brilliant.
Okay, that was a lot. We've covered the 3 main types of conjugation: F+, HFR, and F'. So let's bring it all together in one simple summary, because really understanding the differences between these 3 is absolutely crucial.
This table really sums it all up perfectly. Let's just walk through it. In the basic F+ cross, it's all about spreading the F factor plasmid itself. The recipient becomes a donor. Simple. With an HFR cross, the whole point is to transfer chromosomal genes, but the recipient stays a recipient because it rarely gets the whole F factor. And finally, with F', you kind of get the best of both worlds. You transfer the plasmid and specific bacterial genes, creating a new donor that also carries those extra useful traits. This table right here, this is the key summary to remember.
And that brings us to the big picture. We've looked at all the mechanics, but think about the real-world implications of this. If a single F' plasmid can copy and paste an antibiotic resistance gene across an entire population of bacteria in a hospital, how does that change the way we have to think about fighting superbugs? It means we're not just fighting isolated bacteria. We're up against a massive, interconnected network that is constantly sharing information to survive.
See you in the next video.